and we are just about to start our webinar. We'll just give people a chance to join. We'll just wait for a minute or two, and we will begin our presentation. Okay, so let's begin. Hello and welcome everyone to today's webinar. My name is Marissa Fraser Morera, and I will be moderating today's event. This webinar is titled Barriers and Solutions for Mimicking an In Vivo Extracellular Environment with In Vitro Tools, and will be presented by Connor Gomes, our development scientist here at Pion Inc. Dr. Gomes earned his bachelor's in chemistry from Emmanuel College before earning a doctorate in chemistry at Northeastern University, specializing in the synthesis and characterization of biomaterials. Since coming to Pion, Dr. Gomes has led the development of the artificial extracellular matrices found at the heart of Pion's scissor instrument. Now, also joining us today to assist with our Q&A is Amy Anastasiou, and she is product manager for our subcutaneous injection site simulator, Scissor N3. And again, welcome both to today's presentation. Now, before I hand things over to Connor, I just wanted to let you know we want this webinar to be interactive, and we encourage you to contribute to its success by sending us questions at any time during the presentation. And today, Connor wants to handle questions throughout the presentation, so just send them at any time, and we'll raise them to Connor as he presents. If we still have questions at the end of the presentation, we will continue with a Q&A for about 10 minutes. And if for whatever reason we are overwhelmed with questions and interest, we don't get to answer your question during the live session, we will make sure that we answer them after the presentation via email. Just keep in mind that if you submit a question anonymously, we won't be able to get back to you after the event. So if you want to be safe and make sure that you get an answer to your question, just submit your questions under your name. But again, that's optional. So another thing I wanted to mention, if you do need technical assistance with regards to the Zoom platform, use the chat function and send me a chat, and I'll do my best to help out. The questions need to be submitted with the Q&A widget. Again, just type your question and send it, and we'll be monitoring the questions throughout. And then lastly, I want to mention that we are recording this webinar, and we'll make it available to everyone on our website. And you will also receive an email notification once that recording is processed. Now, without further ado, I will pass things over to Connor. Connor, over to you. Thank you.
Thank you, Marissa. Hello, everyone. I'm Connor Gomes. I'm going to try to not bore you today when we're talking about the barriers and solutions for mimicking an in vivo extracellular environment with in vitro tools. This talk will be very educational in nature. We'll look at a broad view of the subjects, and then as the presentation goes down, we'll narrow the field smaller and smaller and get more complex as we discuss things that come up along this formulation development pathway and the tools that are used within it. As I said, first we'll be speaking about formulation development analytics in general, sort of the pathway and tools used normally along the development route, going from finding your molecule or drug of interest and then going all the way to a formulation months, if not years later. After that, we're going to discuss these biomimetic materials, so synthetic materials that humans make that are included in these types of in vitro assays that are meant to mimic some sort of function within the human body. These are important for these in vitro assays because without mimicking these areas of the body, we can't get the data that we're looking for along the formulation development. We'll then look at the phenomenon that we're actually looking for using these tools, what we're actually looking for upon injection or introduction of a formulation to a biological space, an in vivo space. And lastly, after we understand the sort of the flow through of how these in vitro assays are developed and the materials within them, we will look at the common issues with mimicking the in vivo environment when it comes to the chemistries and architectures that are found there, as well as fitting them to the use inside of an instrument that scientists can interact with and get the data they want out of. So for those of us that are familiar with some formulation development education, there's a very typical pyramid that looks like this, where we see in silico at the bottom and in vivo at the top. This pyramid is normally introduced when talking about formulation development and drug design, where the in silico method at the bottom, in silico taking place in a computer, a simulation platform normally, and the breadth of the width of the pyramid is normally indicative of the amount of drugs that are able to be screened at that point. So in silico, the base of the pyramid, we can scan as many drugs as we possibly want, as many that are in our simulated drug library through these platforms to determine their quality and parameters. As we move up in complexity from in silico to in vitro to ex vivo to in vivo, actually inside of an animal, not only does the cost go up, but I really want to point out about the difficulty of variable control, because the point of this webinar is to talk about the actual instrument development. And so as we move up in this series, our ability as scientists to control the phenomenon going on inside each of these assays gets more difficult and more difficult as we move from computer simulation to a biological animal. And briefly, just to run through these. So the in silico base of our pyramid, where the majority of screening of drugs will be done, involves simulation platforms in which some sort of computer software, statistical software, mathematical calculation is done given an input of details about a drug of interest. So there's a few suites and companies I have shown here in this in silico block. Specifically, we at Pion have our Pion predictor to understand dissolution kinetics, but there are many companies that do mainly one of two things. The first is a simulation of a target. So you have a target, you've identified, what's causing a problem in a disease, whether it's a misfolded protein, whether it's an entire metabolic pathway you have to block, you've identified your target. Crystallographers in the protein field have done a very good job at honing in on these targets, purifying these targets, and crystallizing these targets to create three-dimensional models of these proteins or larger complexes. These software suites will then introduce drugs of interest to this simulation and then see how the two will interact. How those two interact gives people an idea of if they can move forward with that as a drug candidate, or they have to try a different drug. On the other side, you have statistical models of the functions that actually happen inside the human body. So after a formulation is introduced into a human body, there are phenomenon that happen, like with an oral tablet, you have very simple interaction with the mouth, and then as it moves down the trachea into the gastric juices in the stomach, then is passed to the intestinal fluid, and then is absorbed through the body in each of those pathways. All these different pathways, we've done in the past have done a lot of work to understand the kinetics and the forces put on each of these bodily systems onto the molecules that we're introducing. There's a few companies that sum up all of that work into their own statistical models to understand the interactions that drugs will have with the human body without ever even touching a human or an animal. Now that we have an idea of the in silico platform and the reason why you can scan and sort of implement so many drug screens early on in the process for a low cost, we're going to move up to the in vitro methods. This is where the bulk of this presentation will take place. We'll be talking about in vitro methods for the most part. And these are instruments above a computer simulation that involve some sort of glassware, wet chemistry, but they do not introduce any sort of like biological component, especially a whole biological component like you're experimenting inside of an animal. Most of these assays will normally work with a solute or an API of interest. being solubilized inside of a solvent that is supposed to mimic a biological medium that's found within the human body. This is typically done with a simple buffer or more complex buffers where you're actually simulating the gastric or intestinal fluid. Scientists will introduce these formulations to these simulated fluids and monitor how fast this solute is solubilized within its solvent. The scientists then correlate that solubilization with similar solubilization phenomenon and mechanisms that are going to happen with introduction into our human body. A jump up in complexity of that is actually introducing another solution within this chamber to simulate transport across a membrane, but we'll get into that in a little bit. If we're not working with something that can easily be introduced into an overhead stirring apparatus like a tablet or an injection, and maybe a buffer isn't the most opportune environment or appropriate environment to test our pharmaceutical agent within, we can move to different instruments with materials that aren't exactly a buffer, more solid materials like the human body itself, where you can introduce the formulation into either a synthetic skin, a synthetic muscle, in our subcutaneous injection site simulator scissor, and on our end, that is a simulated subcutaneous space. And then instead of a dissolution, normally within a solution, you can see dissolution in diffusion kinetics in a more solid biomimetic material. Now, again, one step up in complexity, therefore one step up in our difficulty to control the variables happening within each of these assays are ex vivo assays. These are a simple example of this is taking an actual body part from an animal, like an eyeball from a cow, en masse, and then plating that eyeball on some sort of surface or device, an introduction of an API into that environment, and then you monitor what happens after. An ex vivo experiment compared to an in vitro will be much more biomimetic, as in an in vitro environment, a lot of the time it's scientists and humans that are trying to create a mimic to an ex vivo sort of assay. So in our subcutaneous injection site simulator, that subcutaneous environment isn't going to have every single biological component that if we went inside a human, grabbed in subcutaneous space to test in, that'd be the most sort of opportune test, but it's very difficult to do, which we'll get into. Or you can have Genoskin is a name of a company that has actually been successful in mass producing these ex vivo human skin models. And so I wanted to mention them because I think it's very impressive that they've been able to do this at scale. And that's sort of like the piece de resistance in the field right now, as far as I know. Lastly, we have the in vivo tests. These are what we think of when we think of animal testing. You have your API or your pharmaceutical agent being introduced into an animal, into a very complex environment where we as scientists have very little control over the phenomena that happens after introduction. And then we monitor what happens to and inside the sites of each of these animals. There are some challenges with these in vivo tests, as they're normally the most sought after tests to do with a pharmaceutical agent, but recent studies with more biological ingredients, biological APIs, monoclonal antibodies, and proteins show that the in vivo models that we've been doing, pig, monkey, and rat models, are showing less and less correct correlation with the in vivo responses that we're seeing in the clinic. So the need to move away from animal products as a testing source, there's sort of a drive for this in the field, and that's really where the in vitro studies that we're going to be talking about during this presentation will really come into play. So trying to cover the gap from using a synthetic material and trying to avoid the complexities of going up to taking biological material directly from a source. So, as I have briefly mentioned, a lot of these in vitro assays have to do with the solubilization of some sort of API or formulation. That API, active pharmaceutical agent, ingredient, is introduced into some sort of biorelevant solution. Examples of this are the simulated gastric and intestinal fluids that I've mentioned already. After introduction of the API into this biorelevant solution, scientists want to monitor how fast this API solubilizes. And normally there are methods to do so where we track how many of these yellow circles spread out into, the solvent. From that, we get these beautiful curves where we can see concentration, the concentration of solute in solution over time as it goes from solid into aqueous. Now, stepping one more up, as I mentioned when we were going through the in vivo processes, sometimes there's an additional component added to these assays. This will be our biomimetic material found here. This biomimetic material, normally simulating a membrane or part of the body in which an API has to pass through, will be placed between our donor chamber, which is the biorelevant solution with the solubilized API, and an acceptor chamber. Your pharmaceutical agent will pass through this biomimetic material and be collected in an acceptor chamber of of the same or a different biorelevant solution. And the rate at which your API is found in the biomimetic material and the acceptor chamber will dictate the in vivo response. So those, the phenomenon and how fast this occurs will then be correlated to the in vivo response that hopefully is seen in the clinic later on. We're going to be focusing on the development of these biomimetic materials. Typically, these materials involve some sort of extracellular components that are found in the human body, which we'll go through in a minute and review those and why they are important to include in these sorts of materials. In addition to the solid materials, there will also be the aqueous buffer that's found in each of these environments. Most of these experiments that I've mentioned already are happening inside an aqueous environment. Within this biomimetic material, although a lot of the time for batch-to-batch reproducibility and cost reasons, cellular constructs are left out of these types of materials, but that's not to say that they are not included. There are catabolic and metabolic pathways that are happening at each either injection or ingestion site that are going to dictate the fate of an injectable. But when it comes to in vitro assays, when we're trying to get a reproducible result from a man-made instrument, cells can add complexities that can sort of raise questions later on. The rational design of these materials have to do with meeting the instrumental requirements. So although we can even if we could perfectly mimic something like it is inside the body, it still has to fit the instruments that we use on everyday bench work. So that's either introducing it into a solid or aqueous medium, reproducibly the same way, no matter what the formulation is every single time. In addition to that, you have to make sure that any phenomenon happening inside of this material can then be observed and analyzed by the scientists that want to probe these formulations. The environments inside of a human body are constantly changing over, and there's constant fluid flow. But when we make a man-made material, whether it be a solution or a hydrogel or a more solid material, there's innate shelf life stability that comes into question. Especially with these softer materials that are supposed to mimic the human body. And so not only are you trying to match all of the characteristics that are important to your assay, you also want to make sure that it can be shipped and reach a scientist and used by the time that the product degrades. And moving forward, we're mainly going to be talking about the rational design of these materials. So what goes into actually thinking about what to mimic, what aspects of the human environment are important to get into your biomimetic material, and what factors are not that important when it comes to the fate of a pharmaceutical, As stated before, we at Pion have a subcutaneous injection site simulator. So I am going to phrase the next part of this talk as sort of probing injectable formulations. That does not mean that any of the concepts that I'm going to mention are not valid when it comes to the solubilization of a tablet or an or an in situ forming gel that's supposed to stay solid in there for a long time. But anyways, so here we see that many different types of mechanisms of injection to introduce formulations to a human body. Each of the distinct areas, the intradermal, intravenous, subcutaneous, and intramuscular that I have on the screen, will have distinct architectures, reactive centers, transport mechanisms, and pharmaceutical delivery mechanisms that fit their architecture and chemistries. First, we have the intradermal injection. This is going to happen with a very small needle, smaller than 26 gauge. So that's about 260 micron inner diameter or lower. And these injections are typically up to or less than half a mil. These injections happen in between the top layer of the skin and the one right under that, the dermis and the epidermis. And so this small volume is quickly introduced to a very thick environment, a very collagenous environment, like we can all look down on our skin and see how tough that is. And so the diffusion out of the injection site is going to largely be dictated by passive diffusion through that tight matrix, and then the uptake by the many circulatory vessels, the tiny circulatory vessels we have in our dermis layers. The next injection, moving down under the skin, is going to be an intravenous injection, excuse me. There's a wide range of needles that this can be accomplished with, from 0.5 millimeter to just over 1 millimeter inner diameter. This will be the fastest uptake of a drug. As we all know, the circulatory system is very quick. And so it's really the uptake of the injection site of an intravenous injection is largely dictated by solubility, aqueous dissolution, and the flow rate actually leaving your application device. So it's a very, although they're both injectables, the intradermal and intravenous routes are going to have very different mechanisms of actions and different considerations when developing in vitro assays to probe them. The subcutaneous injection is going to happen with a very large bore needle, 18 to 30 gauge. That's about 160 microns to just shy of 900 microns in inner diameter. These injections are normally applied very slowly, but can reach volumes from 0.5 mil to 20 mils. And then from very fast uptake to quicker uptake, slower uptake, depending on that volume as well. And because the subcutaneous space is so large, the first initial injection mechanism that's going to happen is that aqueous solubility. And then the subcutaneous space is going to be a very slow diffusion. So you're going to have a lot of interactions with the extracellular matrix proteins that are happening there, structural and solubilized. The slow passive diffusion is going to happen until your formulation interacts with those adipocytes that are present in subcutaneous space. And then lastly, we have our intramuscular injection. The intramuscular injection is going to be done with a needle between around 200 to 500 microns with volumes from 1 to 10 mils, so on the same magnitude as the subcutaneous space, but not as large as intravenous and not as small as intradermal. And there's going to be slow uptake just because of the thick matrix that's present within your muscles, which means, again, just like the subcutaneous space, the primary component of interactions on the solid side are going to be with the ECM protein interactions that are found not only solubilized, but on the outside of those adipocytes. And the, not adipocytes, the fibroblast interactions that are happening in the intramuscular space, excuse me. So although each of these, normally when you're looking for an in vitro assay to probe your formulation, you're going to be looking at instruments that mimic an injection-based mechanism. In the past, the typical experiment is still done with the solubilization of an injection inside a buffer with an overhead stir to understand solubilization. As we move forward and inject larger volumes into more complex areas, the typical solubilization platforms just have less and less correlation with what's happening, especially with things like in situ forming gels and long-acting suspensions that are supposed to stay in their bolus and in their injection site for a longer period of time. And understanding that and understanding how each of these injections are different we look at how to tune a material. So if people are going to be injecting a formulation into an instrument to probe its solubilization parameters and its diffusion parameters, then we have to think about how this sample is being introduced every single time. And most of the time it's going to be through a needle, but each of these needles is going to have a very specific geometry and a very specific force that is put on each of the molecules that is supposed to be your active pharmaceutical ingredient. This is simply because in a needle geometry, you have the fastest molecules moving in the middle of the needle. And then as you move to the wall, you will have a slower and slower molecular movement because of the friction that's felt from the wall of the needle. What this means is that the largest stress, the largest shear that's going to be put on the molecule is going to be right at the wall. And so, excuse me. So what this means is that when we are designing instruments and we are sort of looking at, okay, how do we want our material, our biomimetic material inside our in vitro assay to interact with our delivery device? It's a very hard question to have people use their own custom injection device every time, because every one of these biomimetic materials not only has to withstand bolus injection, so form a nice bolus, but you have to avoid reflux up the needle. And this becomes a different mechanical problem with each different gauge of needle that we're using for an injection. This material not only has to hold the bolus, but it has to allow correct diffusion in 3 dimensions that does not travel back up the needle pathway. Although in a clinic, there is reflux up the needle, but it's been shown that in a good environment in subcutaneous, I believe that's less than 10% of the drug gets lost. And so you still want the bulk of material to fit inside your biomaterial. In addition to mechanically fitting the injection site, we also have to be able to analyze and observe what happens at this injection site. Most commonly, this is done in line, in situ, with some sort of spectroscopic device. IR, UV, Raman, and light scattering and obscuration are the 4 most common that are used. This is because we can develop materials that either forward scatter or don't interact with each of these wavelengths. So why is it a major barrier? Oh, sorry. I apologize about that. The IR, UV, Raman, and light scattering have to interact with not only the material, but also your injectate. And that's actually what the scientists are looking to analyze. And so why does that create a bunch of problems when it comes to designing these biomimetic materials? Well, when we look at the subcutaneous space or the extracellular matrix in general, we find that it's not a neatly organized, clear environment. We have cells that are interacting largely with the extracellular matrix environment. This ECM, extracellular matrix environment, is going to be where the bulk of the interactions happen, and that is where everyone is injecting or introducing their molecule of interest. Within this space, you're going to have solid structures, so that is all the proteins you see. That's the collagen fibers and fibronectins that are interacting directly with the cell surface integrins. And you're going to have GAGs and carbohydrates that are branched and interacting off of each of these fibers. As we know by looking down on our skin, these are not transparent. So scientists have to understand how to get them into a material so that they are there for chemical reactive centers, but not interact with the spectroscopic or any analysis methods that people are using to probe what is happening with their injectate. I've listed a couple the major players here when we talk about these reactive chemical centers, and each of the layers that I previously reviewed will have a different ratio of each of these proteins, although almost every single one of these ingredients will be present throughout. We not only have to worry about the chemical reactivity, but the architecture of the subcutaneous space or the extracellular matrix in general is very important when it comes to the passive diffusion and active diffusion mechanisms present at an injection site. Collagen is the most common protein found throughout our body. It makes up 30%, I think, of our mass, of our dry mass. And so with that in mind, a lot of the time, collagen is introduced as the #1 material that's found in these biomimetic materials that are supposed to mimic injection sites or drug introduction sites in the human body. A lot of the time, they are solubilized in one way or another to achieve materials of certain opacity, and this is solubilized collagen. An issue is that solubilized collagen is not the interactions that your drug is seeing when it is injected inside a human body. Collagen fibers, these are porcine fibers, slight difference from human, but we can get into that later. These strands are not only going to be organized in a very linear manner, but they're also going to have what's called D-banding, which is each of those tiny little ridges that the white arrows are pointing to. Each of those ridges is going to happen at about 670 angstroms. And if you're talking about diffusion through this space, a primary component of that is going to be the available void space in these materials. And this type of D-banding in this microstructure is actually going to come into play, because whether your injection is going through each of those D-bands or it's interacting directly on a collagen fiber is going to have a role in the response you see in the clinical release. So, although collagen architectures have this D-banding in the body, as far as I know, I don't believe the in vitro materials that we've designed so far globally have been able to sort of match this microstructure that's found in the body. And it is one of the major barriers we have at making that jump from making a sort of biomimetic material that matches some characteristics to really making that jump where it's like, okay, we have a material that actually mimics an intramuscular environment because we have this architecture as well as these chemical reactivities. So not only do we have to focus on the microstructure of these fibers, but the macrostructure of where we're found and what layer we're in these injection sites is going to matter, obviously. So if we look at the subcutaneous space, it's going to have largely adipose sites and interstitial void space. There's going to be passive diffusion before some capillary transport after injection from either a cardiovascular or lymphatic, capillary. However, we like to think of the subcutaneous space as filled with fatty tissue and lipophilic molecules. This is a zoomed in image of the outside of an adipocyte. The fat cell will be absolutely a major component in what your injection sees when it's introduced into the subcutaneous site. But there's also a matted mat of collagen that is taking up space before you get to that adipocyte. So even though, like I said before, the subcutaneous, intramuscular, intradermal, and capillaries are going to have different ratios of all of these extracellular ingredients, there's still generally an order of operations that happen after an introduction of a formulation into these spaces. And the interaction with collagen is one of them. And just to bring that point home on how the architecture of collagen matters is whether we have a diseased or healthy state that we're mimicking in these materials. So we have solutions that mimic a fasted and fed state of intestinal fluids that are widely available and widely implemented in these types of experiments. But when it comes to mimicking the solid components of what happens within the body at an injection site, the healthy and diseased states become a little bit of a larger question of sort of ion balance. So we have these are images of muscular tissue, and image A is showing, this is incredible. Sorry, I forgot to reference the work. This is work done out of the University of Denmark, the Niels Bohr Institute, and the University of Copenhagen, where they were able to image a healthy patient heart and then a heart after a stroke occurs. And you can see that, yes, we see in the healthy patient, you have the aligned collagen fibers that we previously viewed with the debanding in a previous slide. Everything is aligned in parallel so that when we look to mimicking this kind of environment, we understand, okay, when we finally get to the point in which we are introducing solid fibers of collagenous material, we need to align them at this angle, at this spacing, in order to achieve the same diffusion kinetics that happen inside this environment. After a stroke or after a disease state, you can see that the linearity of these fibers changes. And that means that you will simply have a different diffusion coefficient simply by the solid architecture difference within the same injection site. And I simply just wanted to point this out because this is a common challenge when it comes to designing these materials as to what state and what geometry we're actually building these solid materials from and what our final goal is at the end. And tuning a material to do both a healthy and disease state is an elusive procedure and an elusive methodology thus far. So just quick last mention, one more barrier, and that is actually what happens when you introduce collagen and structural proteins into an aqueous environment that's supposed to mimic the in vivo. Up here, we have a very typical example of a collagen hydrogel that was formed at ambient temperature with solubilized collagen. Most collagens, when they are solubilized, can exist in a clear solution. And the bulk of publications in science that's put out thus far, the resulting collagen-based hydrogel has been opaque in nature, simply because collagen loves to assemble on itself. It wants to try to form those fibers. It wants to try and form solid materials at neutral pHs. And so this is what the bulk of materials have looked like. As mentioned before, there's a lot of spectroscopic analysis that wants to happen within these materials, especially after you introduce a pharmaceutical agent into them. Since this time, Pion and others have come up with little tricks in order to form solid materials that have a significant collagen concentration that are also optically clear when it comes to these different types of spectroscopic analysis. And now using these types of materials, like the ones found in our scissor, although this is a collagen-based prototype, these types of materials are now used to understand what happens during an injection, after injection, and diffusion away from an injection site. The large takeaways I hope from this talk have been, these biomimetic materials have a need to be implemented in vitro assays moving forward, especially as we move to more complex pharmaceutical agents like biologic material. Not only the chemistries matter with these materials, but also the structural and architecture that dictate that passive diffusion that scientists are looking for, especially with these injectables. Scientists want to know how fast their drug is moving away from an injection site, and we have to make sure that we're tuning those architectures correctly. Not only do we have to fit all of the architectures and chemistries found in the in vivo environment, but we also have to make sure that it can be reproducibly implemented within an instrument, within something that already exists in each of our labs and can fit on a lab bench. And yeah, so I hope you've all enjoyed this talk. I hope it was educational in some capacity. Thank you for paying attention. And if there are any questions, curious or not, complex or simple, I would love to thank them. Thank you very much.
Thanks for that, Connor. Yeah, that was a really interesting overview of the ECM and the challenges that you have when trying to mimic that environment in vitro. As Connor mentioned, the Q&A is open, so please do feel free to add any questions. So we've just got one here. Can collagen be used as in vitro GI tract membrane model?
Yes, eventually, when we talk about the specificity of actually, I didn't mention that. So what's been seen is that there is a difference between animal-based collagen and human-based collagen when it comes to the mechanics that the fibers that they form can produce. And what this tells us down the road on the chemistry side is that more than likely the type of collagen and how the collagen is organized will dictate diffusion through certain membranes, which means if you have a collagenous matrix like the wall of an intestinal membrane, if you are going to probe the holistic chemical reactivities that happen during transport through that membrane, collagen will more than likely have to be involved because there are going to be complexation sites and aggregation sites that happen on the collagen molecule that's present in the intestinal wall or in the stomach wall.
Thanks, Connor.
Yeah, no problem.
We have another one. Why are other extracellular matrix proteins not used as often? Not used as often to create biomimetic materials?
Yes. So the only collagen is the most widely used extracellular matrix protein that's involved in these materials because it's not only available, it's also inexpensive. Out of a lot of animal products, we can get collagen and gelatin. In addition to that, the other extracellular matrix proteins are become very complex when they are in a solid or solubilized form. You can take fibronectin, for example. I actually can go back to the slide. The fibronectin molecule, for example, can absolutely interact with an injectate or a pharmaceutical agent or excipient. But in the extracellular matrix, fibronectin is existing as two very different analogs from one another. One of them is a very small solubilized, I think it's hundreds of nanometer sized sort of sphere. And on the other side, you have an elongated fibronectin molecule that stretches out nanometers and nanometers. And so when you're talking about making a material out of this, out of these proteins, the control that we have been able to achieve so far in material fabrication has not allowed us to control those conformations. So when it comes to fibronectin or the transmembrane integrin cells, scientists just haven't had the time yet to understand how to complex them and control their architecture when we include them in aqueous environments and more solid materials.
Thank you. I think that's actually all our questions. We can leave it a couple of seconds to see if anybody has any more questions. Please feel free to put them into the Q&A tab and we'll answer those. But if there's no more questions, or if anyone wants to ask Connor anything after the event, oh, we've just had another one. How long must the API travel to be vascularly uptaken?
Oh, okay. So that's going to be different in each of the areas that we are injecting within. So in the dermal layers, you have very, very small capillaries. They're running highly vascularized environment. But the vessels are very, very small. So I'm not sure at the actual magnitude off the top of my head, but that compared to an intermuscular or subcutaneous environment where the capillaries are more spread out and they're also much larger, there's going to be two very different answers for these different areas of the body. And so I'm sorry I can't give too more of a specific answer for that, but there will be different distances and different diameters that are traveled through in each different area of the body.
Absolutely. Thank you. Well, I've just got another one coming through. What thickness of collagen should be used as a GI tract membrane? Sorry, I think.
Thickness of collagen is an interesting question. So the thickness of collagen will most likely be, so let me just think really quickly. So the thickness.
I think this was also for GI tract membrane
Yeah,
as well, just to.
yeah. The thickness, again, I do not have a magnitude for you, but where I would start is the diffusion characteristics through the entire membrane wall when there is an inert reaction. And so there are based on the optical properties or material properties that are trying to be matched with the intestinal membrane or the gastric membrane. I would tune the concentration of collagen so that it first matches the diffusion of an inert molecular particle. So if I was mimicking an intestinal wall, I would sort of set up in a hydrogel environment where, because most of the materials that mimic the in vivo environment are hydrogel in nature. And then based on how thick of that material fits inside my instrument, I would match the diffusion speed of an inert molecule like a nanoparticle through the intestinal wall with that, and then I would change the diffusion parameters by adding in collagen to then match up to that passive diffusion. That's where I would start with that one. The thickness, I can say why the thickness is a hard question to answer directly. Based on the concentration of collagen in these biomaterials, you can have vastly different optical and material properties happen. I forget where this study's out of. I apologize to whoever the brilliant scientist is that came up with it. But there was a study where they compressed a 60 mg per mil, dehydrated a 60 mg per mil collagen solution and then pressed it between two glass panes. And that's what they used to mimic the ocular membrane. And so it was a transparent membrane, but it had all biorelevant molecules within it because of that collagen matrix. And so that's kind of why I'm having a problem with this question, because the thickness will have to be tuned based on what you're trying to hit when you're trying to match an intestinal wall or match the gastric membrane. I just think you really need diffusion experiments, passive diffusion experiments to understand where to go with your collagen concentration, because the final properties can be drastically different depending on your manufacturing method of the collagen. So yeah. I'm sorry that was a ranty answer, but yeah.
No, I think that was really interesting. Thank you. We have one more. Since you've talked a lot about injectables, which device from Pion would best mimic the injectable dosage drug release to be used during formulation development?
Yeah, so that's a great question. The scissor platform, here's an image of it here, has the artificial extracellular matrix that I talked about, that is supposed to mimic the subcutaneous space. Now, what happens with this? I definitely should have had a supplemental slide of the scissor instrument, but the normally a needle is placed within this cartridge and an API is injected into a bolus at the top of this cartridge. The phenomena during injection, like aggregation and diffusion, will be able to be seen through this cartridge with cameras and LED optical sensors, opacity sensors. And then as it leaves, you can see the LEDs lighting up the cartridge green in this image. As it leaves that green cartridge, Pion has a fiber optic spectroscopic tool called the Rainbow, where we can measure how much drug is coming out of the artificial ECM over time. And that is what we would correlate to the in vivo response when it comes to drug release and mechanisms that happen after injection that would sort of impede release as well.
Okay, thanks, Connor. And I think there was just a supplementary question to that about asking whether there's any publications or webinars, case studies about that device, which on our website, we have a really good section of the website where you can log in and download resources. We also have all of the other webinars that have been done on YouTube platform as well.
Mhm.
So those can all be accessed there.
Yes, and if there's any specific questions about scissor literature, feel free, pass along questions or specific questions you have about a publication. And we can try to point you in the right direction as far as what scissor publication would be appropriate. Because not all of them that have ever been published can be found on Pion's website. So if there's additional questions, please don't be shy.
Absolutely. With that in mind, I think we're running out of time a little bit. So I'll just say thank you again to Connor and hand back over to Marissa just to close out the webinar. Thanks.
Thank you, Imi. And thanks, Connor, for sharing this very interesting presentation with us. Again, if you missed the introduction, we recorded this webinar and we'll make it available on our website, and we'll share also the link with you via email within 24 hours. So it's usually faster than that. I want to thank again our speakers today for their contributions, and of course all of the participants for being a great audience and making our presentation interactive. As Amy said, be sure to check our website, not only for content related to this presentation, but there's also upcoming webinars and events posted there regularly. So do make sure you check with us regularly. And thanks again for joining us today, and we'll see you next time. Thank you, guys.
Thank you very much, everyone.
Goodbye.